Good evening, everyone, and welcome to Straight Science. Straight Science is an evening presentation series, science presentation series put on by University of Alaska Fairbanks Northwest Campus here in Nome and University of Alaska Fairbanks Alaska Sea Grant here in the office at Northwest Campus. So both Northwest Campus and Alaska Sea Grant, we are public servants of the Bering Strait region. We serve all peoples of the Bering Strait region, which is the homeland and waters of the Inupiaq, St. Lawrence Island Yupik, and Central Yupik peoples. So tonight we've got a very applicable straight science to the region, though it will be focused in the Beaufort Sea, but those whales we're going to be talking about are the same whales that come down here and really in Bering Strait fashion, we would say the whales live here and return here, and they're only visiting up at the north. But hopefully no one from the Beaufort Sea is on because they may have a different opinion. Kathi Lefebvre is no stranger to straight science. She gave us straight science in March of 2022 on HABs and the Arctic food web, and the summer of 2019, when, if you lived here, you would know that was very warm time in the marine environment. Kathi Lefebvre is NOAA, Northwest Fisheries Science Center in Seattle. So she's coming to us later for her than for us, and we're already appreciative of that. And she leads the Wildlife Algal-Toxin Research and Response Network. And the acronym for that is Warren West. With that, I'll turn it over to Kathi. We're so glad you're here to tell us the story of the bowhead whales, the multidisciplinary study to get your results, and to hear what the whales themselves had to tell you in this project you've been working on and leading. Thank you so much.
Thank you. Awesome. It's great to be here. I'm going to share my, you see it?
Yes,
Yep.
It looks great.
Okay, great. Thank you. Okay, yes, I am actually really excited to share this project with you. It's an incredible study that was only made possible by a very close collaboration with the North Slope Borough, the whaling communities there, the bowhead whale health studies. And so I want to explain to you what this study is, who the authors are, and what we learned by using bowhead whales as environmental biosamplers of the food web in the Beaufort Sea. So this is a list of the co-authors on this study, and I really wanted to put this up here to show the multidisciplinary team. So we couldn't have done this without the North Slope Borough and the whaling communities and the harvest and the programs that's up there. This author list shows we have a physiologist, veterinarian, physical modeler, bio and geophysical oceanographer, conservation biologist, algae specialist, a phycologist, Arctic wildlife biologist, toxin chemist, climatologist. This was an incredible team to work with and to make the most out of this data set. So Gay mentioned the Warren West team. This is, again, the Wildlife Algal-Toxins Research and Response Network. We've been operating for, some of you probably already heard of this, but we've been operating for about 15 years and collaborating with peoples from as far north as Beaufort Sea to Southern California. It's the entire west coast of North America, including BC. What we do is we accept samples from stranded and subsistence harvested marine mammals, test for algal toxins. So this provides the surveillance for where these toxins are and what kind of exposures are occurring. We're often involved in analyzing samples during unusual mortality events or any kind of wildlife mortality event. And as we were running this program, we developed all of these really great collaborations that led to the study that I'm going to be talking about. To let you know what toxins we're talking about, we work with pseudonychia species that produce domoic acid, and they're responsible for the human illness known as amnesic shellfish poisoning. And then we work with Alexandrium species, which produce saxitoxin, and though that toxin is responsible for the human illness known as paralytic shellfish poisoning. So these are the two harmful algal bloom species of concern that we work with and the toxins that we test in the food web. I would like to mention here at the beginning that the Alexandrium dinoflagellate also has what's called a cyst stage. That's what that little picture is there, that little circle above where it says cyst. And that's another stage for this species. What happens is the cells can form cysts and drop them to the bottom. They're kind of like seeds, and they can stay in the sediments there until conditions are right, and then they can germinate and seed bloom. So the Alexandrian species have two kind of ways to cause blooms. Cells can be advected in from the south, or they can actually germinate from cyst beds in place. And I'll talk a little bit more about that later. And to top it all off, these algal species occur together, so we can get both of these toxins in the food web. This is an image showing both the Pseudonychia diatoms, which are the look like cigar shaped things, and then the Alexandrium, which are the circular things. So, like most like all phytoplankton in the ocean, it's the base of the food web. Most algae are good. Their primary productivity, they support the whole ecosystem. The species that we're talking about produce these toxins, making them harmful, and that's what we call harmful algal blooms. So filter feeding organisms like zooplankton like krill and copepods, or planktivorous fish, or mollusks like mussels and clams, etc., can filter these algae for food and then accumulate these toxins, which then pass through the food web. And this is a story that's, probably we've all heard multiple times, and we know it's a growing concern in the Arctic. What you may not have seen, earlier in my career, I did a lot of toxicological studies, laboratory studies with controlled exposures using different models like zebrafish or a mouse model. So I wanted to show you what saxitoxin paralysis looks like. This is a zebrafish larvae, and this is something called a touch response. And that's the normal touch response for a larval zebrafish. And the one below is what happens when there is saxitoxin in the water around the larvae. So that just shows you how basically there's no activity. We don't have any videos of marine mammals uh, suffering from saxitoxin poisoning. You can see that it's not a very characteristic thing compared to this image of a sea lion is showing a sea lion that's suffering from domoic acid toxicity. And so it's a much more visual impact. So during our Warren West studies, we were able to focus on the Alaskan coast and the Arctic. And this study was published in 2016 and was a surprise to us. And probably many of you have seen this figure multiple times. It's really been all over. We looked at about 900 stranded or harvested marine mammals from 2004 to 2013. We tested for saxitoxin and domoic acid. Domoic acid is in red, saxitoxin's in purple. We had about 13 different species listed here. And just to sum it all up, we've found that in all regions and in all species, these toxins were detected. Now, this study was not at all designed to say whether these doses that the animals were getting were impacting the animals. It was basically just saying these algal species and these toxins are there in the background, they're in the food web. But we don't know how high. A lot of the concentrations in these animals in their fecal samples or stomach contents were very low. But that is really what raised our concern about the Arctic in particular, because we can see that those toxins are there, at least at low levels. And we were discussing and predicting that the warmer Arctic conditions increase the risks of harmful algal blooms. So, in other words, warmer ocean temperatures and loss of sea ice are more favorable for harmful algal blooms. And we know that because, excuse me, labor- sorry, laboratory studies have shown that warmer ocean conditions, or I'm sorry, warmer waters increase the growth rate of these algal species, and the loss of sea ice also allows more sunlight into the water for growth. So, even though we believe this will happen, how can we quantify that? How can we say that that is really happening? And that's where this study comes in, this incredibly unique opportunity. So, again, this is in collaboration with the North Slope Borough. We received samples of harvested whales through the bowhead whale health studies in at the department, the North Slope Borough with Rafaela Stimmemeyer. This is an example of a fecal sample extraction, and so we had a total of 205 whales sampled between 2004 and 2022. So 19 years of fecal samples. And we're using the whales. Well, so we were actually starting this to just say, hey, how are these toxins a risk to the health of bowhead whales? Are they a risk? Are they exposed? And as we were going along, we're thinking, oh my gosh, we have this very long data set with fecal samples from whales that are filter feeding throughout the Beaufort Sea on zooplankton. So it's a very good sort of sample of what's in the food web. And these were all these animals were from the fall harvest in the Beaufort Sea. And so we had 3 to 19 whales per year over the 19 years. And we could look at yearly prevalences of domoic acid and saxitoxin, which means, so for example, if we had 5 whales that were positive for toxin out of the 10 whales that were tested that year, that would be 50% prevalence. Additionally, we could look at actual toxin concentrations. So, how are these toxin concentrations changing with environmental conditions? So, this is showing the prevalence data. And so if you look at this graph, you can see that at the bottom of the graph on the x-axis, it says domoic acid and saxitoxin. So the green sort of violin plot shows the prevalences were for all 19 years of domoic acid. And the red violin plot shows the prevalences of saxitoxin over all years. And so the important thing to notice here is that over 19 years, at least 40% of the animals tested each year had detectable concentrations of saxitoxin. So there are no years where there's no saxitoxin in the bowhead whales. The domoic acid one shows that there are years where you don't detect the toxin, and that there are many years that are below 40%. So what that is telling us is that saxitoxin is already more prevalent than domoic acid in the Arctic region. And we think we know why that's the case. So, after we look at prevalence, we can look at the actual toxin concentration. So, in this data set, what we're doing is we're taking all 205 whales and we're looking at the toxin concentration for domoic acid and for saxitoxin in each whale. And then we're looking at the accumulated heat flux in the 20 days before each sample was taken. So there are 205 data points. And to try to get an idea about how concentration might be affected, we grouped these animals into low, medium, and high. So the low is the blue line, the medium is the yellow line, and the high is the green line. And so all the animals that had toxin concentrations from 0 to 5 nanograms per gram for domoic acid are in the low group. And then For the medium group, it's 5 to 100, and then for the high group, it's above 100. And it doesn't really matter what these concentrations actually are in numbers. It's just showing that we have three pretty equal numbered groups that we can use to say, how does heat flux impact toxin concentration? And if you look at the figure A, that little map there, the gold star shows where the mooring is located for which the heat flux measurements were made. So heat flux is the heat moving into the Beaufort Sea from the Chukchi. And when you do this model, and this is done by Pegan Lin and Bob Picart, you can see a very clear pattern for both toxins that the warmer the water, which is the accumulated heat flux on the y-axis, the goes with the higher toxin group. So look at that top green line. That's the highest toxin group and has the highest heat flux starting at 20 days out. The low blue line has the lowest heat flux, and then the middle one is the medium one. So it's like a perfect study that you're hoping for when you get your results. This panel below is saxitoxin. And what you notice at the saxitoxin one is that this pattern emerges and then gets kind of fuzzy afterward at 10 days prior. So it's a shorter period of time. That heat flux at 10 days shapes the toxin concentrations. So this clearly shows that warmer water is linked to higher toxin concentrations for both toxins. So then we also have some other measurements that we were able to make, and that is looking at wind speed and sea level pressure for domoic acid and saxitoxin toxin groups. Again, so these are the same groups: low toxin, medium toxin, high toxin. The first column, domoic acid, the second column, saxitoxin. If you look at wind speed, so the top two maps there show, the red shows very strong or high wind speed. So strong winds from in the direction from the Beaufort towards the Chukchi are linked to the low toxin group. Those are the conditions. In the medium group, it's medium wind speeds. And in the high group, it's the lowest wind speeds, meaning the weakest winds in that direction. When those conditions were there, we had the higher toxin concentrations. Now, this is really interesting because it sort of gave us an insight into the potential sources of the cells that are producing these toxins in these regions. So, if you're looking at wind speed, we know that stronger winds from the Beaufort to the Chukchi can inhibit or reduce the flow from the Chukchi to the Beaufort Sea. The weaker winds allow this more movement there. So we believe that the cells, which we can see in some of the other kind of mapping things, are coming from the Chukchi or south up to the Chukchi and into the Beaufort. Another sort of metric we looked at is sea level pressure. So that's the last two columns for domoic acid and saxitoxin, and we see a consistent pattern that fits this mechanism. Higher pressures are at the top. That's the oranger color. The low toxin group is linked to higher pressure. The medium group, medium, and the high group, lower pressure. And conveniently or obviously through the mechanisms of the pressure, the wind is higher with higher pressure and the wind is lower with lower pressure. So we can see sort of a mechanism of movement. We can see that warm water is related to higher toxin concentrations. And then the next metric we could look at is open water. So we looked again at prevalence. And what this figure is showing is that if the years that had 100% prevalence, that means all the animals in that year contained domoic acid, and then the years in which it was less than 100%. And what the bars are showing you is open water area. So there was a statistically significant higher open water area for the group in which there was 100% prevalence compared to when there wasn't 100% prevalence. And this is in June, July, August, and September. So this is another metric that is quantitatively showing what we were predicting, is that more open water would result in higher toxin concentrations or be more favorable for harmful algal blooms. And so, I mean, this is part of what the mechanism, what we're thinking about. The more open water, we looked at the relationship between open water area in June and sea surface temperature in July. And what this figure is showing is that there is a very strong correlation. The more open water in June, the higher the sea surface temperatures in July. So, warmer ocean conditions equal more toxins in the food web. And we're looking at this through prevalence and through concentrations. The higher the heat flux, the higher the concentrations of both saxitoxin and domoic acid. More open water, which is basically the same as saying a loss of sea ice, is equals higher prevalence of both saxitoxin and domoic acid. So, saxitoxin is more prevalent than DA or than domoic acid in the Beaufort Sea food web. So, why is that? So, remember we talked about Alexandrium, which is a saxitoxin producer, has a cyst stage. And if you look at this map to the right there, the cyst map for Alexandrium, that is showing by color scheme concentrations of cysts in what are called cyst beds throughout the Arctic. So the red or orange represents higher cyst concentrations. And our collaborator, Don Anderson, who's been studying Alexandrium and cysts for his entire career, has commented on how these are some of the highest or most dense cyst beds in the world. And if you look up there where it says Western Beaufort Sea cyst bed, that's our sampling site. That's where the heat flux comes in. So that is an additional source of bloom initiation compared to Pseudonychia, which does not have a cyst stage and is going to be relying completely or mostly on advection from the Chukchi Sea. So, so this was a pretty tight story that was only made possible because of this long-term data set and this collaboration and using the bowhead whale as a biosampler. So we were able to quantitatively show that the warming Arctic increases the risks of HABs. And this last sort of set of figures that I want to show you were put together by Rick Toman, also co-author in this paper, showing how sea surface temperatures are warming over since 1900, since records were first taken. So this shows the northern Bering Sea, and the red dots show the 10 warmest years. The blue dots show the 10 coldest years, and then the green line is the estimated average. And what you notice is that after the year 2000 is when all of the warmest years occurred. Similar for the Chukchi Sea, again, 1900 to 2020, the 10 warmest years after the year 2000. And in the Beaufort Sea, a similar trend. There's one outlier there earlier on, but the same upward trajectory. This is also then open water area or sea ice extent. Sorry, this is sea ice extent, which is an indication of open water area. So what you can see is a decline in sea ice extent since 1970 to present, with a pretty dramatic change after the year 2000. So there really appears to be an increased warming rate after the year 2000. And we've been able to show basically in this study that this is increasing the risk or increasing the presence of HABs and their toxins in food webs. So this is, as I said, a collaborative study. This is a short talk because it's a very clear and concise study. And I'm We're very excited to answer any questions and have a good discussion about this. So thank you.
All right. Well, thank you, Kathy. And while everyone's thinking about their questions, this would be a good time to show the speaker a little love in the chat box. It's never easy to be a public speaker, and Kathy's on a later schedule than us even. So thank you very much, Kathy, and we're very appreciative. So with that, we'll throw it open for questions. And if people are still thinking about that, I can ask one right now. And that is, I just wanted to listening, I was learning that, you've got the open water. We know things are warming in the Arctic. And throughout all this, you have high prevalence, so that the bowhead and are more often apt to have a detectable amount of these neurotoxins, both domoic acid, the one that kind of puts you in seizure mode, or saxitoxin, which puts you into sort of the paralytic mode, paralytic shellfish poisoning. For all that you found in your analysis, it was very low in toxins. The number, the poison rate. Was there anything dangerous to the bowhead?
So that's a good question. One of the great things about this study, it's kind of two ways to look at it, but the bowhead whales in this study were used really as biosamplers of the environment, and so we showed mechanistically and quantitatively using biological and oceanographic information how the conditions are impacting toxin levels in the food web. But it doesn't say anything about health impacts to the whales. It doesn't say anything about their exposure levels. We're doing some of that work. We're doing some modeling in our EcoHab study, and we only have like 4 years of data. And we're kind of working with that. And we can make predictions about their exposure rates. And we have in one of our papers looked at saxitoxin because that's the most prevalent toxin there. And we found in our first sets of models from our food web sampling that the toxin levels were not likely getting high enough to impact bowhead whale health yet, but based on them feeding on krill and known concentrations in krill, etc. We did see that walruses had higher exposures, and that's because they're feeding on clams as opposed to krill. So we are not seeing what we call really alarming levels in the feces of these whales. So this study is not saying that whales are being poisoned. It is only saying that the risks and the exposures are related and growing with climate change. And that's what we're saying.
Yeah, thank you. That helps clear it up. There is, I see Sue Moore's hand is up, and go ahead, Sue.
Yeah, thank you, Gay, and thank you, Kathy. It's really great to see this study in no small part because of your team. So I'm listening tonight with bowheads on my mind, as they always are, but also with gray whales on my mind because we have increasing concerns with gray whales. And so my question is, those cysts that are, I believe, in the sediment or on the sediment, gray whales could be sucking those up. And I'm wondering, are the cysts themselves poisonous? In other words, would they increase a gray whale's saxitoxin levels and perhaps impact their health?
That's an excellent question. So we know, yes, cysts they do contain toxins. That's a lot of work that's being done in various places, and especially at Woods Hole Oceanographic Institution. Um, there's Courtney. Oh, uh, there's a study by Courtney. I can't remember her last name right now, who was looking at cysts moving through gooey ducts, etc. So there's a possibility that the cysts can go through the digestive tract without being broken.
Yep.
So it's possible that the cysts don't have a huge influence in terms of toxin exposure to filter feeding shellfish or even maybe bottom feeding whales. So that is not 100% clear right now. It's
Okay.
certainly worth thinking about. One thing is that if cysts are germinating and bowhead whales, I mean, and gray whales are eating from the bottom, then yes, there can be a contribution there. We know from from many different studies that those animals that eat closer to like the shellfish, the benthic
Yeah.
feeders have higher exposure risks.
Okay. Good. Thank you. I'll probably follow up with you.
Yeah,
Thank you. Great talk, Kathi.
thank you.
All right, Rick, go ahead. Your hand is up. The floor is yours.
Great. Thanks very much, Kathi. Such a clear explanation, even I could understand it, which is no easy task for a climate guy. I guess really one of the things I've been wondering is, given the scope of this project, really focused, using this wonderful historic record provided by North Slope Borough and hunters and wildlife, the veterinary folks there. Can we use this study as part of the arsenal to make the case on why we collectively need to be monitoring for this, for these toxins on a more systematic level? As you mentioned, in other species that are widely used in the Bering Strait regions, there's reason to think the concentrations would be higher. So can we use this paper as yet another piece of evidence of we need to do better at monitoring this in real time for people's health? Thank you.
Yes, I think absolutely. I think that's the biggest value of this paper, having the 19-year data set, because we know it makes common sense what we're saying we think is happening. And people, well, you can't prove it. You can't prove it. We did. Now, this is showing that this is happening. We kind of predicted this, but harmful algal blooms globally are a major impact of climate change. These increases globally. It's becoming there's a massive one going on in Australia right now that they've not had before. It's a huge thing. So it is very clear from this study, and it can be used to show that we have to have a monitoring system, because it's going to be in places that it hasn't been before, and it's going to be in higher concentrations. And one of the things we're doing with our next EcoHab grant is to really focus on subsistence species other than the whales. I mean, we'll still be working with whales and walruses, but we're going to be looking at the entire food web with more focus.
Excellent. Thank you.
Right. And there was in the chat, Kathi and I was texting, and I think maybe I've tried to put it there also in the chat. There's a question regarding the names of the different toxins, what they do, and who produces them. So you might want to take a look at what I wrote, make sure I got it right, and then reiterate.
Okay, I could, maybe you could send out that slide to everyone, that when that first slide shows the toxins, the species, and how they work. I was just thinking a pictorial thing would be.
Sure. Can you share your presentation, and then you
Yes.
could zip right to it maybe? I don't have a copy of it, so.
Let's see.
Because they do very different things,
Yes, I think
those
this
different
is,
toxins, and that's an important thing for for a lot of us who are looking at animals and looking at animal behaviors to get signals as to what might be going on. So if you could go over that again, that would be awesome.
yes. Okay. So this is great, because I would like to expand on this. So the domoic acid is one of the toxins we're talking about. It's produced by these diatoms, Pseudonychia species, and it's an excitotoxin. So what domoic acid does is it binds to glutamate receptors, which are the normal neural transmitters in the mammalian system. It's also in birds, in most systems. And what it does is it overexcites the nerves. So it causes seizures, confusion. It can cause brain damage, memory loss, and obviously in the worst cases, death. And that was showing that sea lion having a seizure. So those are very obvious excitotoxic symptoms. And we see this every year in Southern California. The first event was documented in 1998. I was actually a first year graduate student and involved in that study, showing that this toxin could impact marine mammals. And since that time, dozens to hundreds of animals are impacted every year in the California region. This last year was the largest event so far. Thousands of animals impacted by domoic acid coming on beaches. It started earlier. It started in February, which has never started that early before, and lasted a really long time. The marine mammal stranding networks and the communities were exhausted, the animal care centers, etc. So domoic acid is really visible. Saxitoxin is a sodium channel blocker, which just means it blocks nerve function. So instead of overexciting nerves, saxitoxin blocks the nerve. So you cannot have an action potential, which is what you need for movement. So that causes paralysis. It's not going to be very visible. One of the things we're thinking, we know that saxitoxin impacts marine mammals. Saxitoxin is actually a little bit more potent than domoic acid. So it is likely that animals are impacted. There's only been a few studies indicating that saxitoxin may have impacted some marine mammals until this last year. And maybe we can do another straight science on that event. But in this Pribilof Islands, there were northern fur seals that we were able to show were exposed to high levels of saxitoxin, and they had washed ashore. They were dead, so we didn't, see any symptoms at all. But the high toxin level proved that it can cause the death of marine mammals, and most likely, if you're exposed to saxitoxin out in the ocean, you're going to stop breathing. So you're not even really going to drown. You're going to mostly just, it would be like a suffocation for the animal. That would make it less visible for these events. So I'm not saying that these events are happening all the time, but I'm saying they were much easier to miss a saxitoxin poisoning event because it causes paralysis versus a domoic acid poisoning event, which causes the visible seizures. Does that make sense?
Makes sense to me, but if I could try to add on to or for my understanding, and so I'm glad you're here. So my understanding would be for domoic acid produced by the algae called Pseudonychia. That one, and we don't see much of it. That one excites your nerves, And so you'll see that rocking repetitive motion in a sea lion or whatever animal is impacted, has eaten too much of this, and this is in its bloodstream. And it'll arch its neck, or it'll do these repetitive movements or be shaking. Its body is too excited. The nerves are too excited, and it cannot control its movement. And it will go into that seizure mode.
Yeah.
Then you've got the saxitoxin, and that's made by the algae Alexandrium, which we know we had not up in the Beaufort Sea for the bowhead study, but down here we certainly had a good dose of it in 2022, the summer and late summer of 2022. And for that one, my understanding is, and it's so good you're the toxic expert, it's blocking, when you say sodium channel blocker, it's blocking, like I'm making a fist. My brain's just saying make a fist, and I'm making a fist. And it's that messaging from the brain to your nerves. That's the opposite one. And it goes completely, slows it way down until you're going to be paralytic shellfish poisoning, right? So the animal, or person, I suppose, but in this case, I'm not going to talk about people, but the animal, like a walrus or a bearded seal, something that may eat something that has too much of it in it, it will go it's having trouble because it's telling its body to do something, and its body is not able, the muscles are not able to receive that signal at the time it's got the poison in the bloodstream. It's going to be working its way out if it can, peeing, pooping, throwing up, whatever it can do. But in the case of, we had a case of an animal, and we have such expertise in the Bering Strait region with all the animals' behaviors. So in 2017, we had that Diomede, at Diomede, there was a walrus that came out of the water. People called it over. That's not surprising. But what was surprising, it came right out of the water, and then it wouldn't move. It just put its head down. And you don't know what it would take to make a walrus react to saxitoxin, but it was interesting that that particular walrus was dispatched, and the contents of its feces were analyzed by you, and it was 5 times over the seafood safety limit. And that animal that had the saxitoxin, which causes paralytic shellfish poisoning. And the bad part of that, if you really have a bad case of it, your diaphragm is going to be paralyzed, and your brain's saying, draw a breath, right? Draw a breath. And it won't be able to do that. Have I got that?
Yeah, that, yes. And just, if, I mean, if you guys are interested, I love neurobiology. I'm not a neurobiologist, but I have some knowledge, and the sodium channels are in your nerves, and those are the channels that open and close and let ions in and out. And you need to have that happen at a certain level before you'll get what's called an action potential, which sends the signal and sends out the neurotransmitters. So it's really just, it's almost like it's just a functional blockage of that whole system.
Right. So I'm thinking, open your hand, right? It's happening. But if you had a real bad dose of that saxitoxin, you'd be thinking, open your hand. And you may not be able to do that. Would that be correct?
Yes, you would, yeah, not be able to do anything.
Right. And that's something to look for, I think, when you see super tired, something to really note, super tired animals, or and I'm not just saying walrus, it could be any kind of animal, mammal or bird, that's approachable if you're in the field harvesting, or on ice. A lot of our animals are on land, or not on land, but they're not in the water at certain times of year. So you really get a way to, you have a nice long time to look at them, look at their behaviors. And there's such expertise in this region. It's really great to give. So thank you very much for giving that kind of how that works and the different things to look for. I think everybody here, and I wouldn't say everybody in the region, but certainly lots of people in the region would note, because we have to rely on, people have to rely on their own knowledge for keeping their family safe from disease and sickness with our wildlife. And we don't have regular monitoring. So people are very keen and have a great knowledge of the animals' normal behaviors, many, many different kinds of animals. So please note that if you are out in the field in the Bering Strait region or further north, to note that. Those are
Yeah,
things
and
to look for.
it's valuable to start noting that, and then we have to link it to other factors, because that's what's hard about paralysis is it's not really a defined thing, and exhaustion and fatigue and all those things could be caused by other things as well.
Maybe you
So
got hit in the head by a boat. You wouldn't know what it is, right? You might be seizing because you've got a brain injury, and we just wouldn't know. So just a notable thing, huh?
hey, I see that there's a Courtney on here.
Yes, there is. I saw that earlier.
Courtney, what's
Courtney, you're being called out. If you
your
want,
last name?
you don't have to tune in or get off, but if you want to, there you go. There's a
Oh,
visitor.
hi.
Hi.
Okay,
Hi.
I was just wondering if you were the cis study person that I was.
Oh, not me. No.
Hi.
Hello.
Thank you,
Thank
Courtney.
you.
All right. Any other questions regarding the study in the Beaufort Sea? I've
Hey,
got a question. Oh, go
I
ahead,
do.
Byers. Go
Bye.
ahead.
Hey, can you hear me?
Yes,
Yes.
sir.
Hey, this is Bivers. Gay, I want to thank you for reminding me about this meeting here. Um, And Kathy, thank you so much. All the explanations you've done and the ice conditions over the years is very easy to understand. And another person I want to thank is Rick Doman, who's always given us information in our region about the ice conditions, the water conditions, and also the temperature. I want to thank all three of you. You know, you guys are very valuable people in our subsistence way of life. I've been hunting since the 60s and 70s, and you know, I'm still active hunting to this day. But you know, what Kathy put on that graph with the 70s, 60s, and 70s, we had a lot of ice conditions that were pretty thick, and you know, they lasted till June, July, and I'm just surprised that ice conditions from 2000 to this day just spiked up to where, you know, a lot of people say we have ice here, we have ice there. Well, as a subsistence hunter, we don't call that ice. Um, you know, to everybody in the Western world thinks ice is ice, but to us subsistence hunters, it's not the type of ice we're looking for. Um, but I'm curious in regards to the bowhead whale having this from being the bottom feeder. And my concern also is with the walrus is also a bottom feeder, eating clams, and same thing with a bearded seal.
Yeah.
And as subsistence providers, we've always passed on or shared all our food to everybody, family members and friends and out of state and stuff like that. And I've said it before in the past that I'm always concerned about sharing my subsistence food because of what's happening to our subsistence food now in the ocean. Should I be more concerned now that it's more abundant with what's going on?
Yeah, that is the $1,000,000 question, right? That's what we all want to know. I can say that you are correct in that walruses and bearded seals, which consume the clams, etc., have a higher exposure risks to themselves. So the animals themselves have a higher risk of having health impacts. But what we're finding when we look at exposed marine mammals at their various tissues, that we don't see high concentrations of the toxins in muscle or blubber. We see very, very little. So I feel pretty confident at this point to be able to say things like muscle and blubber are not going to be a human health risk for consumption in terms of algal toxins. We're continuing to look, though, at organs like kidney and liver to see, oh, could we get, because that's how things are excreted, could we get toxin levels that could be dangerous? And then for the other types of subsistence foods, that's what we're going to be doing in this next study, is sort of looking at, and that is the question. We know that shellfish accumulate these toxins, and yes, it's happening more often. What is going to be the answer in the long term is a way to be able to test the harvested food to see if it's safe to eat. And that's a complex issue that's difficult to get going in remote areas, or in all the remote areas. So I guess the answer to your question is that there's some good news in that the marine mammal tissues are not a concern when I'm talking about muscle and blubber.
Okay.
But that, yes, we do need to be concerned about increasing toxins in various things that have traditionally maybe not had. And
Can I jump in for
yeah,
one second too? So this is a really good, thank you, Bivers, for bringing that up, and thank you for your kind words earlier. This is really important, Kathy, because it's the walrus I did mention, Bivers, that Diomede walrus of 2017, where we knew what killed it. That was a person killed it, but the unuk was fresh, the feces was fresh. And actually, just by circumstance, other tissues from that walrus were also sent in. So Kathi was able to look at, okay, wow, this was high for saxitoxin. And we had muscle, we had some blubber, I think. And so for the skin and the blubber of the walrus and the meat, that saxitoxin was either not detectable or extremely low, while you had it really high in the feces. My concern, Kathi, is we have the intestines where the toxin transfer is occurring, the stomach, the liver, and kidney, which are trying to purge that toxin out. And those are all mightily high priority subsistence foods. And so those organs, is that is something maybe that we'll be getting to
Yes, that
in
is
our
what
new
I've been
project?
working on for a while. We're accumulating a data set. That's what I really think could be incredibly valuable. Can you ever get concentrations that are dangerous for human health in liver, kidney, and intestinal, tissues?
And
We're
stomach.
working we're working on that. Stomach contents are a different story, but so we're working on that, and the thing that's taking so long is that we have to catch an animal that has high toxin in the feces to be able to prove that it was eating really toxic food, and therefore we can look through the system. But I do want to say, from a toxicological standpoint, as a toxicologist, it is not likely that there's going to be very, very high concentrations outside of the digestive tract, because the animal is going to be impaired before it can accumulate too high levels in the rest of the system. So you get high exposure to food, you take up some saxitoxin, and then you're going to stop eating, or the animal is going to stop eating. So there's all kinds of things that we're sort of trying to put together, and hopefully within the next year we'll be able to put out a document that has a lot of data to show what we think about the organs and those various parts.
I also want to share with you guys in regards to subsistence harvesting is the bearded seal, the walrus, the minke whale,
Very
uh,
well.
gray whale, and the bowhead whale. There are times when they come up they wash up on the beach, and we do harvest the meat of all of those marine mammals. And for your information, with both the bearded seal and the walrus, and I'm pretty sure they do with bowhead whale as well. We take the intestines, we take the liver, we take the heart, we consume that. Um, to me, right now, uh, I think back 20, 25 years ago, 30 years ago, we had Fish and Wildlife Service that we as subsistence hunters provided everything for testing. And I had mentioned that to Joel Garlick a couple of years ago in regards when I was on that research boat or on that the boat studying the walrus. I
Yeah.
said, why can't we go back to that? Because we as subsistence hunters, we also give you the internal organs of the animals to get tested. Would that be a beneficial thing for any communities that do hunt and collect these samples and get them sent in with Fish and Wildlife Service? Because honestly, this is scary. Just listening to you guys talk about this.
Yeah.
Um,
We've been getting the samples, the suite of tissues from harvested walruses from Gamble and Savunga as we can. This year we only got 13 animals, and we're analyzing them right now. So if we have toxin concentrations in the feces, which I predict we will, we'll be able to provide a table of all 13 animals and all the tissues and see what kinds of concentrations we're getting in the organs.
would
Um,
that be would that be beneficial with subsistence hunters as well? Since we hunt the walrus and the bearded seal, if there is feces visible, I mean, I wouldn't mind getting samples for you guys. Um, give that sample as well too. If it's gonna help you and everybody else. You know what I mean?
Yes. Yes, absolutely. And that is, yes, we are, we like to take samples, and Gay's been instrumental in getting us those. So any subsistence harvested samples that you want to send in. Now, I do have to say, we are not able to do this for like deciding on whether you could eat something, because we don't have that authority, and we don't have the staffing to do it that fast. But for the study part, like we're talking about here, like, do you ever see toxin in these various organs? We will take any samples from walruses or other marine mammals, and analyze those, and continue to increase this data set to share with you.
Well, yes, and we also not harvest, but get the clams from the walrus too as well. We also eat those right off from the stomach. We're bringing it home and share with everybody. And is it, to me, it sounds like it's a red flag all around for us subsistence harvesters.
Yeah.
Uh, you know what I mean?
Yeah.
I just don't want to give it to anybody. And
Yeah.
oh man,
That's the hard part of that, because the walruses are really acting as a harvest tool, right? If you're eating
yes,
from the bottom of the ocean. So those clams in the guts of walruses carry the same risk as other shellfish harvested in places. And the state of Alaska does not have a good solution to monitoring shellfish that's used for subsistence. And Gay, you wanted to just add something?
Yeah. So Bivers makes a very good point, and that is we need right now the current levels of monitoring and surveillance for the people's needs here are unacceptable. I mean, that's just that's the answer. It's unacceptable, Bivers, the way that we have this situation and whatnot. Kathi is a researcher trying to figure out, like, walrus or, bowhead. And probably it's going to be a little bit easier with a walrus because it's smaller to figure out. She's trying to figure out, well, if you have this much saxitoxin in the in the unc of the feces, how much goes through into the intestine? And she's got a longer timeline. She's trying to make some of the first trail for everybody on how this works in an animal. And stop me if I've got this wrong, Kathi. So any samples would be great for the purposes of understanding how this moves in a walrus, how this moves in a gray whale, so forth, how it moves in a bird. For the monitoring for subsistence for marine mammals, it's going to be difficult because of the time it takes to get a sample in.
yes,
She's
yes, it
running it
is.
in a lab, and unfortunately, the state of Alaska doesn't do, I don't believe they do of marine mammals, the feces. And so anyway, we've got that. Then we have what you brought up was the invertebrates, the upa, the tunicate,
Yeah,
and
yes,
all the tunicates, right? All the different kinds of tunicates. And then we have crab and we have clam. Now those, they're washing up on the beach. You're getting them
yes,
from
they
the
are.
stomach. Yep, you're getting them from the stomach. That is actually quicker to find out. And that's more, both for the researcher, great to know what is the walrus exposed to, but also what are we exposed to? And so if you want to send in clams from the stomach, those can go to a lab quicker and they can do a faster turnaround. Still not going to be perfect. What they do in Kodiak and Southeast Alaska is they do this harvest and hold. They'll harvest their clam. They live with this trouble all the time. It's new to us, but they're not using the marine mammal component the way we do. So that's different for us. So the clams and the upa, we can get those tested. You would harvest them, you could put them in a Ziploc bag and say whatever date, and send some in to both Nome Eskimo community, Emma Pate, or myself here at the Sea Grant office at Northwest Campus, or with the Norton Sound Health Corporation Office of Environmental Health. That's Austin Amasek, Madeline Kowalik, and that crew. We're all teamed up together. Chuck Manadlik over at Coeric Subsistence. So we're all teamed up, and those kind of samples would be fantastic to get, both clams from the stomach as well. And by that, I don't, it would be nice, Kathi, to know if it's the whole clam. If you catch a walrus right when he's been eating, there's a whole lot of the clam in there, no shell per se,
Yes.
big
Yeah.
clam, but there's a lot of other materials in there, like the stomach full of the algae. You see the green liquid when you're in the walrus stomach. That's all that algae being released from all the other animals it's been eating, all the other invertebrates it's been eating for dinner or breakfast. So those kind of, even a test tube full of that green liquid in the stomach, I would think would be, just dip plastic test tube, and I can get, that's an easy one. If you have liquid in there, it would probably give some signal. So things you find on the beach, we can deal with that. But it does take time. So that's that harvest and hold. It's very frustrating. It is unacceptable, but it is our reality here. And that is tough to live with right now, but that's what we have to do. And everyone's working to try to make it better. Know that if someone is eating something like walrus liver or upa, and they
Yeah.
start feeling sick at the table, or crab, because maybe people on here don't know, but the whole crab is eaten except for the intestine type part. So if you're feeling sick at the table, and this has happened, people have gotten to feel sick, they stopped immediately and they switched foods. So they went to something completely different. They box that food in, they go to the clinic, which I highly recommend. If someone's eating something that doesn't feel well, go to the clinic. And they would send that material, that crab or that upa, whatever you were eating, and get it tested. That is a high priority item if there's a direct human health threat. And the state of Alaska Department of Human Health will fast track that, and you will get a turnaround on those food items right away, because they want to know, do we have somebody suffering? So there is a little bit more backup plan or priority if someone is actively not feeling well. And that's about the only comfort I can kind of give on this. And we have utilized that, and the amount of saxitoxin in the food that was sent in was very, very low. It was not their problem. They were feeling much better by the next, they had a nice sleep and they were feeling much better and everything was fine. And it had nothing to do with paralytic shellfish poisoning, but boy, howdy, did people react quickly. I would also mention that the teamwork that is with Rick and his maps, and Kathi with all her expertise in the toxins, and everyone here, Nome Eskimo, Norton Sound Health Corporation, we're using Rick Toman's sea surface temperature maps right now, piping those right in to show the hospital here, be advised, the water's very hot, and they are giving their medical staff kind of the thought, if someone came in and presenting with the symptoms of either domoic acid or more likely paralytic shellfish poisoning from the saxitoxin, our medical providers are already getting that. And I'm sure I don't know, but those messages, however they communicate to their staff in the clinics, that's really powerful stuff. So it's another example of everybody, teamwork makes the dream work. Everybody coming together with whatever expertise or whatever service you can provide. It is, again, unacceptable, but we're all coming together as best we can in that Bering Strait fashion, if that helps.
Yes, I would love to share with all the subsistence owners as much as I could in Nome, and also both Edwin's here right now, and same thing with Sivung and Gamble in regards to what's going on with our food. Um, I just don't want to, you know, be sorry for myself or somebody that I give share the food to, and something drastic happens. I don't want that to happen.
Yeah.
So, is there any way or anything that any subsistence hunters from the Bering Strait region all the way up to Barrow in regards to, something like this, and give them information that this is a possibility? Because I'm
Oh,
just, I'm just, you know, I don't feel right about this. I don't feel like right about sharing, but that's
Yeah.
my concern.
Yeah. So what I heard from Kathi for the marine mammals, and stop me, Kathi, if this is not correct, but you would see in the case of the saxitoxin paralytic shellfish poisoning, you would probably see the behavior change before it really incorporates into the organs, or we don't know that part, or
Don't
before
know
it
that yet.
incorporates in. Okay, so you would, but you feel pretty confident that if you see I'm just make up a bearded seal, and he's lying on the ice, and you could kind of walk up to him. If, well, one, you probably wouldn't shoot it because you'd think something was wrong with it, and that's probably true, whatever it was. It could be other things why he's not walking or running away. But you're saying you would probably see the effects of the poison, like saxitoxin, making that animal tired before the meat, blubber, and skin were affected.
Yeah, I think I can safely say that the meat and the blubber will not contain toxins that are at a worrisome level. It just won't accumulate there like that for these particular toxins because they're what are called water soluble.
Does that help? So that still doesn't answer the question of
Doesn't answer the
kidney
question.
and intestine and or stomach. Stomach, Kathi, is an organ eaten here in the region. Seals, bearded seal up at Shishmaref, people will prepare stomach, put it on the rack.
And we're working on that. We have some of those tissues, we're doing things like how are they prepared? Can you rinse them? Because inside the stomach there can be lots of toxin. But if you're eating at the stomach lining, if you rinse out what's in it, then it's most likely, I don't know. That's what we're going to be testing.
Yeah, you don't want to say too
Yeah.
go out on a limb too far, because people here are really. But I would reiterate that any samples that are provided from marine mammals or other wildlife, Kathi can look at those. It will just take time. The more time than you have to, consume and process and gift the foods you have. For the other foods like upa, like crab, clams, little fish that wash up on the beach, all those things and worms that wash up on the beach, all those things that are consumed in that comprehensive use of the marine ecosystem that we are as a region so brilliant at, so educated to be able to do all that. Those can come in and there's a quicker turnaround time because we can send those in state. So those kind of things, and I'd like to see monitoring, but we only have, thank goodness, we have the research like Kathy's able to bring, but that's a pretty tentative, that's a grant funded effort. The state of Alaska and the federal government who looks after these animals for us, it would be, they're having their own issues right now with funding and reorganization, but boy, howdy, do we need services provided out here. You highlight that very, very well. So thank you for bringing that up. I wish I had a better answer, but at this time, know that everyone's trying the best we can. And even the water being monitored, which we're doing weekly in Nome, and we are also getting volunteers from the Norton Sound Economic Development Corporation is volunteering to, as their tenders are out at sea by St. Lawrence Island or doing the run to Unalakleet, they're taking water samples and we're sending those in. And then Norton Sound Health Corporation is actually taking those water samples and using them under a microscope right now. I'm sorry I'm talking so much, Kathy, but so they're looking real time, near real time, to see if some of these algae types that Kathy's talking about, that Pseudonychia and Alexandrium, are in our waters. It's not enough to make anything safe, but people are doing the best we can, and we're all banding together on that. I hope that helps.
Well, again, Kathy, thank you so much for this information. Gay's been our valuable person in our Norton Sound region. She's the go-to person for everything, anything in regards to birds, fish, and marine mammals. She's definitely a valuable asset for our region and also for Rick. Keep doing what you're doing. You know, as subsistence hunters are always looking at your your database or your weather conditions, water temperature. Those are really valuable to us too, Rick. And I appreciate you for doing that all the time. Um,
You
and
bet.
thank you
Byron.
for listening. Appreciate that.
You humble us, Bivers. Thank you very much. With that, Kathy, I was blabbing away there. It's not my street science, it's yours. So any last thoughts or words on these kind of concerns and where we're going in the future? You might want to mention this. You have a new,
Yes,
can you
we
talk about that a little bit?
we've talked about pretty much everything. I just will reiterate that we are pushing forward in the next grant to really focus on the subsistence foods, that are not traditionally looked at for algal toxins. So the tunicates and various other things. And we're going to be doing some studies to look at how long they retain toxin and do they depurate it and various things like that. So hopefully we'll be having a lot more information on subsistence foods, but there's still going to be the need for the monitoring. And that I know that's sort of in process. That's what people are trying to work on within the communities themselves. So thank you. So it's been a real honor to be here. I appreciate all of you. This is a pretty amazing environment and system, and it's kind of a scary time.
And actually, I will say that Kathy will be in Nome. Looks like we're still planning, but she will be in Nome for a community meeting here. That will be, and it will also be not just in Nome, but it'll be here at Northwest Campus, and that'll be. We're just in the planning stages of getting her to come, so people can have a longer conversation. October 6th and 7th looks like those will be the dates. She will be here with many others that are experts in the research end of harmful algal bloom toxins. And I can't wait because you can come on down, Bivers, and get in a chair and get at the table and they'll answer your questions and mine too, because I've got a ton of them like you.
Yes, I'll definitely be there. For sure. Appreciate that info too.
You bet. All right. With that, I think any other last thoughts or questions? We'll call it a night. The next Strait Science will be on September 28th, and it'll be about the Sekuliak and the work that they're doing right now. So Sekuliak left on over the weekend, Sunday. Is that right? Saturday or Sunday? Sunday. And they left recently. And they are going in the northern Bering Sea, and then they'll be up into the Chukchi. They'll be back in Nome. The ship will be at the dock on the 29th. And they got a whole new crew than the group that was at Gamble. And so there'll be a Strait Science on August 28th from the ship itself. So they don't have time while they're here, but they have the internet power on Sekuliak to do their Strait Science from the ship. So we're going to try that and see if we can get that real time what they've been up to and what they've seen. And that'll be on the 28th. So thank you all for coming. Kathi, if you can stay on 1 second more, that'd be great. And we'll see you. Everybody have a good night. Thank you so much for coming.